Today, Ruochen, who is my fifth-year PhD student, who is going to graduate this summer, is going to present our perspective. It's a perspective article we recently wrote regarding the debate about zeros in single-cell RNA-seq data. There are some controversy about whether these zeros are actually informative or simply noise, or whether we should use those zeros in our analysis or simply remove them in our analysis. So Ruochen is going to offer our perspectives on this. And please note that we're still refining our article. So any advice from the audience today will be greatly appreciated. So please go ahead, Ruochen.
Okay, yeah, thank you, Jessica, for the introduction. So today I want to present our perspective paper, Zeros in Single-Cell RNA-seq Data, Good or Bad, How to Embrace or Tackle Zeros in Single-Cell RNA-seq Data Analysis. So this is collaboration with our lab mates, with my lab mates Tianyi and Dongyuan, and under supervision of Jessica. Okay. So in this talk, I will cover like 5 aspects. So the first is the origin of the zeros in single-cell RNA-seq data, short as sRNA-seq data. And I want to clarify some of the zero-related terminologies commonly used in the literature. And I will discuss about the debate on the zero-inflation modeling and how non-biological zeros could affect the downstream analysis. So we did a simulation study based on this. And finally, I want to talk about when the researchers carry out the single-cell RNA-seq analysis, whether they should choose to use the original data or the imputed data or the binarized data. Okay, so here, this figure is an overview of on the left-hand side is the single-cell sequencing kind of workflow, and on the right-hand side is how each step could potentially lead to zeros. And so here you can see here we have 3 types of zeros indicated: the biological zeros, the technical zeros, and the sampling zeros. So basically, the biological zeros are corresponding to like if there's no existence of mRNAs in the cell, then this is what we call the biological zero. And if there will be no cDNA load into the sequencing library, so and if we have the infinite sequencing depth, there's a large number of reads in total, and if it's a technical zero, then it will remain as a technical zero, because if no cDNAs will be loaded into the sequencing library, no matter how we increase that reads in total, we will still have a technical zero. On the other hand, if the cDNA has been synthesized and when it didn't quite go through the PCR or IVT amplification step, or like even after the amplification, there are still very few cDNAs in the sequencing library, then due to the random sampling of the sequencing total, we could get the sampling zeros. And if all of this doesn't happen, we would have a non-negative or greater than 0 expression value. So this is just an overview, and in the following slides, I will refer to this figure again and show how the biological zeros, technical zeros, and sampling zeros can be reflected from this workflow over here. Okay, so first I want to talk about the biological zero. So a biological zero is defined as the true absence of a gene's transcripts or messenger RNAs in a cell. So the first reason is that many genes are actually unexpressed in a cell. So cells of distinct types have different genes expressed. It's a fact that results in the diversity of cell types. And the second reason is that many genes undergo a bursty process of transcription. A gene may switches between the active and inactive states, and its transcription only occurs during the active states. And the existence of mRNAs in a cell also depends on the transcription rate and degradation rate. So here in this figure, we can see in the biological system, the RNA polymerase could bind to the DNA, and if it does bind to the DNA, it will begin the mRNA synthesis. So in the first column over here, if the RNA polymerase did not bind to the DNA, then there will be no mRNA synthesized, and it will finally result in the biological zero at the end. And on the other hand, even if the RNA polymerase did bind to the DNA, and the mRNA has been synthesized. However, the mRNA could still go through the degradation step. And if at the when we take the samples or take a snapshot of the mRNAs in the cell, if there's no mRNA existed in the cell, it could also lead to a biological zero. So those are the two causes plausible for the biological zeros generation inside the biological system. Okay, so here is a figure showing that how the what we call the bursty transcription. So the gene could transfer between the inactive and active states with the rate Ka and Ki. And when the gene is at its active state, it could begin the the mRNA synthesis and generate the mRNAs. And with the rate delta, it goes through the mRNA degradation step, and the mRNA could be consumed. So here the panel B shows that like if we keep the SM and delta fixed and just change the parameter Ka and Ki, over time what the expression level should look like. And you can see that, for example, in the first figure, we can see that there are some time points when we will have zero expression values. And so if we look at one gene over time, we can also think of it as if we have a lot of genes with the same parameter and we take a snapshot of them, it will have the same kind of distribution. So those zeros due to the bursty transcription will also be the biological zeros, even if we observe a bimodal distribution over here. Okay, and unlike the biological zeros, the non-biological zeros refer to the zero expression measurements of the genes that have transcripts in a cell. So the first type of non-biological zero is what we call the technical zeros. So this is, we think, one possible reason is the imperfect mRNA capturing efficiency in the reverse transcription step from mRNA to cDNA. And the second type of the non-biological zero is what we call sampling zeros. So sampling zeros occur due to the constraint in the sequencing depth. If there are too few cDNAs in the sequencing library, it could potentially lead to the sampling zeros. So here is in the third column, we show how the failure in the cDNA synthesis could lead to technical zeros. So basically, if there is mRNA existed in the cell, then we will, so it will be if we finally observe a zero here, then it will be a non-biological zero. So this course here is a failure in the cDNA synthesis. And if there's no cDNA, then it will, after the PCR IVT amplification, it could not have a non-zero value. And the final result we get is the technical zero. Okay, and then the next is what we call the sampling zeros. So if a gene has too few cDNAs in the sequencing library, it may have a sampling zero. The reason is due to there's a total reason, the reason total has a limitation. So basically we have a sequencing depth that constrains how many reads we can allocate to each gene. And so the gene expression, the sum of the gene expressions across all genes in a cell should have a limitation. And so there could be two reasons lead to the sampling zeros. The first is that there are too few cDNAs pre-amplification. And the second reason is potentially the inefficiency in the cDNA amplification. So there are two major approaches currently used in single-cell RNA-seq amplification. They are called the PCR or IVT. So the PCR amplifies cDNA non-linearly or exponentially, while IVT amplifies cDNA linearly. Although it seems more appealing that the IVT will not change the relative abundance, like the relative proportion or ratio between those genes' expression values, the PCR is still more popular because the IVT needs more cDNA input to be initialized. In other words, the PCR can amplify the gene expressions, even the cDNA input is lower. And there are also sources of the amplification bias that could potentially distort the final ratio between the gene expressions. So those sources could be the cDNA sequence and structures, and the design of cell barcodes, adapters, and primers. And in some protocols, it needs to go through around 15 to 18 cycles of amplification, and this could potentially increase in the amplification bias. So as a result, the cDNA copy numbers in a sequencing library may not reflect the actual proportions of cDNAs before the amplification. Okay, so I summarized the sampling zeros over here. So after the cDNA has been synthesized successfully, if it doesn't go through the or if it has an amplification bias or didn't go through the PCR or IVT amplification, it could potentially lead to a sampling zero. And if there are very few mRNA at the start point, and after the cDNA synthesis, although it successfully goes through this step, but there are still very few cDNAs. And after the PCR or IVT amplification, the relative number is still of the cDNA is still very small. Then in the reads allocation step, it could also lead to a sampling zero. So basically, the sampling zero are like kind of random because if you increase the sequencing depth, for example, to infinite, then it will completely eliminate the sampling zeros over here. And as I have mentioned, if all of these situations does not happen, and we have many mRNAs in the starting point and many cDNAs at the end, and it could lead to the non-zero expression value. Okay, next I want to jump to the next section. So we want to clarify some of the commonly used terms, but they may be ambiguous in the literature. So the first term is called dropouts or dropout. So this term is actually quite widely used, and it was first introduced in the SCDE paper. So in their paper, they describe dropouts, says that dropout describes zero gene expression for genes that show moderate or high expressions in only a proportion of cells. And then the literature following this paper, they use the dropouts to describe, some use them to describe the non-biological zeros, and some use this term to describe the non-biological zeros or low counts, and even some literature used it to describe all the zeros in the single-cell RNA-seq data. And there's a paper that used the dropout as a subjective to describe the existence of many zeros. And on the other hand, there's another term called exact zeros. So it's also, we think it's also ambiguous because some literature use it to describe the larger proportion of zeros in single-cell RNA-seq data than in the bulk RNA-seq data. Some use them to describe the non-biological zeros, and some use them to describe the additional zeros that cannot be explained by the negative binomial model. So in our previous discussion, we try to separate the zeros into biological and non-biological, and related to the sequencing technology. So we think like if we can categorize the zeros into biological and non-biological, then this is kind of a biological concept. However, if we look at this description, the additional zeros that cannot be explained by negative binomial model, so this is actually a statistical concept. So we think it's better to not use them like simultaneously, but to clearly separate the statistical concepts from the biological concepts. So there's another term called zero-inflation, and it's a pure statistical concept that depends on specified model, and it describes the proportion of zeros exists what is expected under the specified model. It could be either biological or non-biological. So because it's a statistical concept, we should not confuse it with the biological concept, such as treating the zeros as biological or non-biological. And then zero-inflated models describe the mixture probabilistic models with two components, a point mass at 0, and a common distribution. So here is we will present an example using 4 count distributions. We will compare the Poisson with zero-inflated Poisson, the negative binomial versus the zero-inflated negative binomial. So in panel A here, we show the 4 distributions with parameters. And in panel B, we show that if we have the same mean, what the 4 distribution should look like. So a direct observation is that if we compare the Poisson with zero-inflated Poisson with a point mass, it can explain a larger proportion of zeros given the same mean. And if we compare the negative binomial with zero-inflated negative binomial using a 0 point mass on the left-hand side, will also enable us to describe a larger proportion of zeros. And the interesting thing is that if we compare C and D, although the zero-inflated Poisson is a zero-inflated model because it has a 0 point mass, the proportion of zeros explained by the zero-inflated Poisson is actually smaller than the zero proportion explained by the negative binomial in this case. So if we change the parameterization, it could lead to different results. However, we can observe in this case, the zero-inflated Poisson does not necessarily explain more zeros comparing to the negative binomial distribution. So the point we want to make here is that the zero inflation is a statistical concept, and when the researchers try to use them, they should specify what model they are considering and what the zero-inflated model they are considering. Okay, so then there's regarding the debate on the zero-inflated modeling, I want to first introduce the concept of unique molecular identifier, or UMI. So basically, each input cDNA molecule has a unique tag, and using the UMI is able to correct for the amplification bias, and it's not available for full-length protocol until recently. So in our study, we include 2 UMI protocols, 10x Genomics and DropSeq, and 1 non-UMI protocol, SmartSeq2. So one question or one debate currently have is, should researchers use zero-inflated modeling on single-cell RNA-seq data? So a short answer is that zero-inflated models are more appropriate for non-UMI data, where the non-zero inflated models are good enough to capture the variance and a high proportion of zeros in UMI data. So why is that the case? Here on real data, we have plotted the cell library size, the number of cells, the number of genes detected. And we can see that for the non-UMI data, the SmartSeq2 per cell, it has a larger cell library size or sequencing depth. However, it includes a smaller number of cells, and the non-UMI data can detect a larger number of genes, basically the genes with non-zero values. And then we use the zero, like in the previous 4 models, the Poisson, the IP, NB, and the ZIMP, we applied them to the real data, and we found that for the UMI data, the non-zero inflated models are good enough to capture the variation and the proportion of zeros. However, for the non-UMI data, for SmartSeq2 data, zero inflated model is more appropriate for at least 50% of the for around 50% of the genes. And we further plot what the distribution, fit distribution look like to the gene expression. So here, the first 2 panels show the UMI data, and the last panel shows the fitted curve for the non-UMI data. And we use the 4 genes across different protocols. And we can see that for the UMI data, the non-zero inflated model has a pretty good fit, and for the non-UMI data, the first 2 genes has been fitted using the non-zero inflated model, where the last 2 genes are fitted with the zero inflated model. And we generally observe that the fitted curve in the non-UMI data does not fit the data as well as what has been the model fitting in the UMI data, just from this figure. And so here, if we use the zero-inflated model, it uses a zero mass to describe the larger proportion of zeros cannot be explained by just plainly the NB or Poisson model. Okay, then we further want to see how when we introduce the non-biological zeros, how they will affect the downstream analysis. So here we have introduced 5 different masking schemes, and so due to time limitation, I will just point out that for the random mask, it's commonly used in literature like about the single-cell RNA-seq imputation methods. Many of the methods benchmark their method, for example, using simulated data, and they introduce zeros, just randomly introduce zeros by setting, for example, with some probability, like each non-zero values has the equal probability of being masked to 0. And here we think it's maybe not the case, because there are literature evidence showing that for the genes with lower expression values, it tends to have more zeros. So when we design our method and do the simulation, we could consider a variety of different masking methods and evaluate our results to see whether they are robust, to those different masking schemes. So in our performance simulation, we try to see the effect of non-biological zeros on one cell level analysis, namely the cell type clustering, and the one gene level analysis, the D gene identification. And we can directly observe that the cell level analysis, the cell type clustering is much more robust comparing to the gene level analysis. So the ARI remains at 1, means that we did the perfect clustering on the simulated data. Even we have added we changed the 70% of the non-zero values to zeros, and we still get a perfect clustering result. However, for the D gene identification, it's in most of the cases, it performance decrease as we introduce more and more zeros. And so here we use 2 very popular single cell analysis framework. The one is the Monaco 3 and the other is Surat. And we can see their difference in performance. And we also observe that using the different masking schemes, it do affect the, for example, here the D gene identification results. Okay, so in the last part, I want to introduce how people treated pre-processed data or their input data before their analysis. So here there are 3 types of input data: the original data versus imputed data versus binarized counts. So the most commonly used approach is just direct modeling on the original counts. And one clear advantage of this is that the original counts are not biased by any data pre-processing steps. So we want to separate our discussion for the tool developers or the bioinformatic tool developers in specific, and also for the tool users, usually when people want to use those bioinformatic tools to make discoveries. So for tool developers, they should always go for the direct modeling approach, so because it has the least effect on the data of the pre-processing. And however, for the tool users, a practical concern is that some of the existing bioinformatics tools do not work well with the original data. So they could use imputation or binarization as alternative to make discoveries. So the modeling on the imputed data is another very commonly used technology approach in single-cell RNA-seq analyzing. And so one advantage is that it's a practical data pre-processing step. And the disadvantage is that it could introduce false signals and it could reduce the biological variation as shown by some benchmark papers. And for tool users, the imputed data has better agreement with the biological knowledge as shown by some of the imputation method paper. And the imputation builds a bridge that connects the sparse single-cell RNA-seq data to many powerful tools designed for the non-sparse data. So the tool users, if they did not find a good method of bioinformatics tools that have already been developed, they can use the imputation as a middle step and use the tools they think they are maybe more reason to produce more reasonable results. And there's a current new view on the single-cell RNA-seq data. So there's a paper recently published that the binarized data can be used for cell type clustering. It can achieve a result as good as the if we use the original counts. So because as we've shown that the cell type clustering, the cell level analysis is actually not so much affected by the number of zeros. And so maybe the binarized data could still capture some of the variation in the single cell data. And on the other hand, there are some bioinformatic tools that utilize the zero or non-zero information in their modeling, and they argue that they achieve a very good result. And however, a disadvantage we think is that it's unsuitable for the gene level analysis, because if we just binarize the data, let's say if we do the DE gene analysis, then the only thing we can do is to compare the different proportion of zeros for two genes under two conditions. So it may lose a lot of information. So for the tool developers, we believe that using binarized counts in addition to original counts for developing new tools could maybe boost the performance of the bioinformatics tools. And for tool users, the binary counts can be used for exploratory data analysis, because there are some methods or tools that specifically designed for the binarized counts, and the running analysis on binarized counts may be more maybe less time consuming and more efficient. So for the future directions, we think in the field is better. We could sort of ways to better distinguish the non-biological zeros from the biological zeros. For example, if we can incorporate like this RNA sequencing, the sequence of the mRNAs or genes into our modeling, we could see if there are correlations between those non-biological zeros and the sequence structures. And the second is we think there is need for systematic benchmarking of using the original imputed and the binarized counts in single-cell RNA-seq data to show like which input data is actually doing a better job for the downstream analysis. And the third is that we think more transparent and reproducible bioinformatics tools is needed due to the complexity in the single-cell RNA-seq data, because in the single-cell RNA-seq data analysis, there are so many steps, including the normalization, the clustering, and a dimensional reduction, clustering, and all those steps. So the bioinformatics tools developed should have transparent and reproducible codes like for easy understand and reproduction. So with that, I want to thank my collaborator Tian and Dongyuan in our group and my advisor Jessica for her mentoring. Yeah, thank you, and I'm open to questions.